Let's start with the basics. What is meiosis? Meiosis is a type of cell division that transforms one diploid cell, which has two sets of chromosomes, one from each parent, into four daughter cells, with half the number of chromosomes as a parent cell, like creating sperm or egg cells. This is an important part of the life cycle since it maintains the same number of chromosomes, assuming no mutations, and creates genetic variation, which is a key factor in evolution adaptations. There are different types of diploid chromosome numbers in a cell, such as 2n equals 4, 2n equals 6, 2n equals 8, etc. These numbers describe the diploid state and the haploid number of the particular cell. We will be focusing on the 2n equals 6 cell, where the haploid number is 3, so there are 3 pairs of chromosomes. 1, 2, 3. For you all to understand this process more simply, we decided to depict this procedure through a dance performance. In this analogy, a dance performance with 12 dancers represents meiosis. Each dancer is a chromatid, and dancers pair up to represent chromosomes. The performance illustrates how chromosomes replicate, pair, exchange genetic material, line up, separate, and ultimately form 4 unique gametes. Meiosis 1 separates homologous pairs, and meiosis 2 separates sister chromatids, depicting the whole process of meiosis. By the end of this video, through the stop motion of the beads and the simple analogy we created with Lego human figures, hopefully you can understand the complete process of meiosis in a fun and painless way.
Before meiosis begins, the cell goes through interphase, which includes G1, S phase, and G2. First, in the G1 phase, the cell grows, synthesizing proteins and making the building blocks needed for DNA replication. In the S phase, DNA replication occurs, creating 2 identical sister chromatids for each chromosome. Additionally, the centrosomes also duplicate. Finally, in the G2 phase, the cell grows even more, where it synthesizes proteins and checks to make sure that DNA is replicated properly before the process of meiosis.
Now for the interphase part of the analogy. Before the performance begins, the 12 dancers are backstage getting ready and pairing up for the first dance. They put on their costumes, practice a few steps, and make sure everything's prepared. This is like interphase, where the cell grows, replicates its DNA, and prepares for division.
Earlier in prophase 1, chromosomes condense and become visible. Homologous chromosomes pair up, forming tetrads. The chromosomes have been duplicated and the sister chromatids are held together with cohesin proteins, which form a ring around the center of the X. Then the homologs line up relatively to the same height, and this is when the process of crossing over starts. This process started with the DNA molecules of 2 non-sister chromatids being broken in very precisely coordinated points. Then, a structure called the synaptimal complex is formed, which holds the homologs together, and this association is called a synapsis. The DNA breaks from its original chromatid and switches with the non-sister chromatid, producing crossovers. The points at which this crossing over occurred are called chiasmata, and can be noticed because the DNA is still connected at those points, while it may be disconnected at other points. On the other hand, the chromosomes contain genetic information called genes, which are inherited from parents, and different versions of the same gene on each chromosome are called alleles. Ultimately, prophase 1 allows homologous chromosomes to exchange pieces of DNA and create new genetic combinations, increasing genetic diversity, ensuring that the future genetic gametes will carry the unique combination of traits. Later in prophase 1, the nuclear membrane or envelope that holds the chromosomes inside it dissolves. This is also when the spindle fibers extend outward from the centriole. During metaphase 1, homologous pairs line up along the equator of the cell, also called the metaphase plate. This equator is located perpendicular to the spindle pulling force. The chromosome's orientation is random, illustrating Mendel's law of independent assortment. Each pair's position determines which chromosome will go into which gamete. Both the chromatids and the homolog are connected to a kinetochore, which is a protein that is on the centromere or center of the homolog and accounts as a handle for the spindle fibers to pull toward the centrioles.
Now it's time to represent metaphase 1 through our stop motion. Now for the metaphase part of the analogy. The dancers pairs line up in a straight line across the middle of the stage, perpendicular to the audience. This represents metaphase 1 because just as the dancers line up in an organized way, homologous chromosomes line up along the metaphase plate in preparation for separation.
In anaphase 1, homologous chromosomes are pulled to the opposite side of the cell by spindle fibers, while sister chromatids remain attached at the centromere. This is why the chromosome number is reduced by half, producing haploid cells after the first division.
Now it's time to represent anaphase 1 through our stop motion. Time for the analogy again. The spotlight guide half the dentures pairs to the left and half the right, splitting the pairs evenly. This represents anaphase 1, because just as the dancers pair are pulled apart together, homologous chromosomes are pulled to opposite poles of the cell by spindle fibers.
The setup for telophase includes one of the chromosomes for each homologous pair as separate poles. However, the sister chromatids still stay together, but are not identical anymore because of the crossing over in prophase 1. The spindle fibers then dissolve and disappear while the nuclear membrane reforms, except this time there are two membranes, one on each side of the cell. Cytokinesis splits the cytoplasm, forming two separate haploid cells. There is a step after that happens sometimes, but not often, but as soon as this process is done, the chromosomes start to uncondense, going back to the chromatin form. However, this process is halted quickly due to meiosis II usually being the immediate next step for the two haploid cells.
Now it's time to represent telophase and cytokinesis 1 through our stop motion. Now, back to the analogy. Now the dancers form two separate groups on opposite sides of the stage, and the curtains close briefly. Each group now has 6 dancers, but the original pairs stay together.
So after the diploid cell splits into two haploid cells, they undergo the process of meiosis II. In meiosis II, the haploid cells repeat close to the same process as meiosis I, just with a few changes. Now we have 3 X-shaped chromosomes in each haploid cell, and we enter prophase II. A key difference in meiosis II compared to meiosis I is that there is no such interphase for the haploid cells to prepare for meiosis again, and there's no DNA duplication again before meiosis II starts. Prophase II, same as in prophase I, the nuclear membrane breaks down throughout a process driven by proteins that trigger chemical reactions in the nucleus, which slowly break it down. And yet again, the spindle fibers attached to the centrioles appear on opposite sides of the cell. Later in prophase II, the microtubules from the centrioles and spindle apparatus then stretch out toward the kinetochores at each centromere of the chromosome. This phase is shorter than prophase I because no new crossing over occurs. Now it's time to represent prophase II through our stop motion.
Now, back to the analogy. A new dance begins. The curtains open again and each group of 6 dancers step onto the stage. The pairs stay the same as in meiosis I, and the spotlights, spindle fibers, shine on them as they prepare to move. This represents prophase II, when the nuclear envelope breaks down and the chromosomes are free to move. For the rest of the analogy, we will be only focusing on one group of 6, but the same concepts apply to the rest of the dancers.
So between the prophase and the metaphase, there is this transition state that's the prometaphase II. So as referenced in the name, this process happens as a cell transitions from prophase II to metaphase II. In this process, the spindle fibers actually attach to the kinetochores. The chromosomes also start to slowly move toward the metaphase plate, which is located on the other axis of the cell, perpendicular to the spindle's pulling direction. It is important to note that the spindle fibers connect to the chromosome from both poles, not just one side. So in metaphase II, just like in metaphase I, the chromosomes line up at the equator, also called the metaphase plate. Due to the crossing over that happens in prophase II, the two sister chromatids of each chromosome don't have the same gene sequence, and thus they aren't genetically identical. In addition, the kinetochores of the two sister chromatids that make up the chromosome are still attached while they are lined up at the equator. The sister chromatids are arranged randomly at the equator, just like the chromosomes are in metaphase during meiosis I. This precise arrangement ensures that each gamete receives one copy of each chromosome.
Now, back to the analogy. The dancer pairs line up in the middle of the stage again, ready to be split apart. This represents metaphase II, where sister chromatids line up along the metaphase plate in preparation for separation.
Anaphase II is when sister chromatids separate and move to opposite poles of the cell. They separate by the proteins being broken down at the centromere connecting the sister chromatids, which allows the chromatids to separate. The chromatids then get moved to opposite poles via the spindle fiber. The chromatids then become individual chromosomes and they will reside in a separate haploid gamete.
Now, back to the analogy. The spotlights guide individual dancers from each pair to opposite sides of the stage, separating them completely. This represents anaphase 2, when sister chromatids are pulled apart, unlike in meiosis 1, where the pairs stay together.
During the final phase of meiosis, there are two new nuclear membranes, temporarily, that form on opposite sides of the metaphase plate around the chromosomes. The spindle fibers begin to disappear and cytokinesis occurs. The actual chromosomes start to decondense as the nuclear envelope forms and go back to their very first form in an interphase called chromatin. This happens because chromatin in its less condensed or open form is necessary for most circular or cellular functions, particularly gene expression such as transcription and DNA replication. The highly condensed X-shaped chromosomes seen during meiosis and mitosis are essentially inactive and cannot perform these functions. The meiotic division of one of these parent cells produces 4 distinct daughter haploid cells. The 4 daughter cells are also distinct from one another and from the parent cell.
Now, back to the analogy. Finally, the dancers split into 4 unique groups, each performing their own small routine, and the curtains closed for the last time. This represents telophase 2 and cytokinesis 2, producing 4 unique gametes, each with distinct set of chromosomes.
Even though the process of meiosis is precise, there's still the possibility of errors occurring. When these errors occur, the gametes that are produced don't have the correct number of chromosomes, or the chromosomes aren't in the right arrangement. One of these errors leads to non-disjunction, which occurs when the chromosomes don't separate properly during meiosis. This typically happens at anaphase 1, since the homologous chromosomes should move to opposite sides, but through this error, some stay on the same side. As a result, one gamete receives 1 more chromosome, while another gamete receives 1 fewer chromosome. This is also seen in anaphase 2, where the sister chromatids don't separate and move together in one gamete. One example of this is Down syndrome, where there's an extra copy of chromosome 21, also known as trisomy 21, disrupting normal brain activity and body development. Another error is deletion, where one part of the chromosome is lost because of unequal crossover, which creates missing genes. This typically occurs at prophase 1. One example is the Cri-du-Chat syndrome, where a piece of chromosome 5 is lost, which causes physical and intellectual issues such as crying like a cat in toddlers. So duplication occurs when a part of the chromosome is copied twice as a result of unequal crossover, which creates extra copies of genetic material. This also occurs at prophase 1. One example is Charcot-Marie-Tooth disease type 1A, also known as CMT1A, which is a duplication in chromosome 17. It is an inherited neurological disorder that leads to muscle weakness and sensory loss. So the error of inversion occurs when a part of the chromosome breaks off and flips its position, hence why it's called inversion, and reattaches backwards, changing the gene order. This occurs at prophase 1. One example is hemophilia A due to the inversion in the F8 gene, which creates a bleeding disorder that shows a defect in clotting. So translocation occurs when a part of the chromosome breaks off and attaches to a non-homologous chromosome, which puts genes in the wrong place. This also occurs at prophase 1. One example is chronic myelogenous leukemia, also known as CML for short, where parts of chromosomes 9 and 22 swap, and where they're also called a Philadelphia chromosome, leading to an uncontrolled increase in white blood cell growth.